States in '68, and I was at Yale University for about 20 plus years. I did my undergraduate medical graduate degree, I was a faculty there. And then I was recruited at Tufts to start a department of pharmacology, and very quickly became very interested in inflammation, which is another way of saying immunity. We'll describe that shortly, and especially brain inflammation. So I'm not a psychiatrist, I'm not a neurologist, I'm not a pediatrician, but as you will see, a lot of the work that I'll present is very relevant, although from a different perspective than what we heard before. I will actually give you a lot of basic information, and I hope you will not get tired. I'll try to make it as simple as it can be. That's the only way I can actually go from A to B, and then try to go back to A or make some sense of what we can do. So I will sort of present the work, just the work was being done, both in the laboratory and in the clinic, with or without patients, and why certain things may or may not be helping, and where things can actually fill in the gap, some of the things that we've already heard. We all know the kind of problems autistic children have. But I will start actually by expanding on just a few of them. Let me just interrupt for a second. Denise, do you know why that last import's still up there? By any chance? I can see those words up there. It'd be good. But I want to stress the fact that there are many different symptoms, many of them having actually inflammation. So there's brain inflammation that looked at brains of children with autism, and they found out that certain molecules that we know are involved in inflammation. Try the next one.
It's not
Okay,
there.
that's right. Are there, as we heard earlier, that doesn't mean to say that the presence of molecules like IL-6 or interleukin-6 or tumor necrosis factor are the beginning of the end. It might be just symptoms, but they're there. There is no question that there is disruption of the protective blood-brain barrier. You may recall or you know that the brain literally lives on sugar. It cannot burn lipid, it cannot burn protein. It's very protective, so if it breaks down, all kinds of noxious things can get into the brain. And we know that the blood-brain barrier is disrupted because about anywhere between 20 and 60% of autistic children have antibodies against their own brain proteins. In order for that to happen, that means the immune system, which never gets into the brain, the white blood cells, got into the brain, recognized the proteins of the brain as foreign, because they're not supposed to get into the brain, and start attacking the brain. So we call them auto-antibodies, so they shouldn't be there. Many of the children that have the ability to at least speak tell us that they have sort of brain fog. Other diseases have that. They cannot quite concentrate, whether it's eye contact or not paying attention to directions or what have you. But to us, it was very telling that a lot of children had food allergies, food intolerance, GI complaints, and I'll get back to that very shortly. And a lot of children actually had skin, I call them eruptions, let's say skin allergic problems. And you'll see why I try to avoid the word allergy shortly. But we've heard so much about genes and genes that have been discovered in autism and what on earth that means. And in fact, 90% of the funding agencies, whether it's Simon's Foundation, Autism Speaks, the National Institute of Health, have been directing the money towards discovering actually potential genes that might explain autism. However, there has been no disease really, and certainly not autism, that has explained or has been explained by genes. In certain cases of cancer, we know that there might be susceptibility or higher susceptibility if you have one or another particular gene mutation. But certainly in psychiatric diseases, there's never been any gene that has explained a particular disease. And even though about 3 years ago there was a well-publicized study made out of Denmark on identical twins, which study showed that about 2/3 of the identical twins will develop autism. A study was published out of California about 6 months ago with 3 times as many identical twins children, and it showed exactly the opposite, that only about 1/3 could be explained by genetic associations and not the rest. So it leaves us wondering what else might be going on. Of course, all the parents have been struggling with this. The point is, can we pinpoint anything to which we can sort of start hanging facts on and then sort of build a theory that might lead us somewhere? So this paper was published in a very good journal called Neuron just recently. And in just 2 pages, it listed all the different mutations that have been discovered in autism. And what struck us is that a mutation in many cases showed up both here, here, and here. You can't quite see it. But the mutation had to do with a regulatory molecule that is called mTOR, M-T-O-R. It doesn't mean anything to you, and it's too complicated to explain. But in those cases, there was high association with autism in children, in those particular cases. The reason why we looked at all of these genes is because we were interested in inflammation. So we were trying to figure out, is there any gene that might at least relate to what we were doing, so that one might be able to put 2 things together, and one might be susceptibility, the other, which I will tell you, might be sort of the trigger of the susceptibility that might lead to something. And very good reviews have been written recently indicating that signaling, as we call it, of this particular molecule, has to do with memory and attention and sociability, etc. And they've done this both in animals and in humans. So it has stood so far the test of science, whatever that means. There's at least one gene mutation that seems to be prevalent in certain kids with autism, in certain animals with autism. And why are we interested in it? It's because both we and other colleagues recently showed that if this mTOR pathway is activated, it activates a certain type of immune cell called the mast cell. And you will hear plenty about the mast cell from me. So, on the one hand, we have a mutation, then we have a phenotype, meaning the way patients present, and now we have something that has to do with the immune system. So, obviously, the immune system is not working properly if this pathway is actually activated. So what are mast cells? Mast cells were discovered by a German scientist in 1887, and he called them mast cell because mast comes from the word mast, breast, in Greek again, and he thought that these cells were feeding other cells, just like the breast feeds the child. And the reason he thought that is because these cells have about 500 such little granules inside each cell, and each granule has about 50 or so molecules. But the only thing we've known about this cell is since about 1947, when it was discovered that if this cell gets activated, it releases histamine, therefore allergic reactions. Yet, the only drug we have to date relating to these cells, or to allergy for that matter, is antihistamines. How to block histamine after it has been released, yet these cells release another 49 or so other molecules, which we don't really understand what they do, and we certainly don't have any way to block them. And what was fascinating to us, because we've been studying the cell for almost 40 years, 40 years since I was an early student, 20 years actively in the laboratory, is that in addition to the trigger that is well known in allergy, which is called immunoglobulin E. So this is one of the immunoglobulins that circulates in the body. It homes and sits specifically on about 500,000 specialized sites on the surface called receptors. And if you're re-exposed to whatever was the trigger for you to make the Ig antibody, we call them antigens. It could be pollen, it could be whatever. Then 2 or more of these specialized areas move closer together, and they start a cascade of reactions that leads to what we call degranulation. So if you're unlikely to be allergic to penicillin, someone shoots you up with penicillin, this cell will explode like a hand grenade, release everything. It will constrict your bronchi, drop your blood pressure, blah, 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 and you most likely will die. That's why you carry an EpiPen. However, we were the first to show that this cell type can release individual molecules without ever undergoing this explosive degranulation, which means you will never know it if you just took a piece of tissue and you send it to pathology. Because in pathology or histology, you look for this cell to degranulate, and it's obvious. if it releases only one molecule at a